So limitations include the fact that while we see evidence of anti-tumor activity with checkpoint inhibitors like PD-1 inhibitors, they tend to work less than half the time in the vast majority of tumors. And so what's important to do is see, now that we know we have an agent that exerts some activity in such a broad spectrum of cancer types, what can we do to increase that response rate from less than 50% to 100%? That's our challenge. GITR, the GITR molecule, is a member of the immune checkpoint family, and the rationale would be if you can actually stimulate that receptor or hyperstimulate it, it's a different class of antibodies than, say, antibodies which block tumor activity. You're actually trying to exert more activity. And the point of doing this is to deplete activity of T cells called T regulatory cells that exist in the tumor microenvironment and are sort of hampering the immune system's response to cancer. So if we are able to do that in conjunction with a molecule like a PD-1 inhibitor, you might be able to reinvigorate the T cell response against the cancer in a way that you can't do with PD-1 alone. So the monotherapy agent did not show evidence of anti-tumor activity that was significant. In fact, there were not any responses in the dose escalation cohort. Now, that's not actually surprising when you look at this in context of other molecules similar to the one tested that have been reported. Monotherapy doesn't appear to be effective. When you combine the agent with pembrolizumab, a response rate of 69% was seen in a very, very small sample size of 13 patients. I think those results need to be interpreted with some caution because pembrolizumab on its own certainly has evidence of activity that could be seen, and the activity we saw could be explained just by the pembrolizumab. Nonetheless, 69% is higher than what we're used to seeing, so I think some further interrogation into the combination and the molecular rationale as to why we saw what we saw would be very important in understanding how this molecule works. So I have not been privy to what plans will be from the study sponsor, but from my perspective, I would think it would be very important to understand the biology of the compound by looking at tumor and blood samples from patients collected as part of the study, and really nail down what the pharmacodynamic effects of this drug are. It's hard because there may not be a clearly defined target, so to speak, but understanding what the actual effects of the drug are on the tumor and within the peripheral blood of patients, I think is important to understanding whether it should be developed further. So we're testing a number of receptors that I think of as cousins within a family, and those include molecules like OX40 or 4-1BB or CD40. I think we're still in very early stages of understanding what the role of agonizing these molecules may be, and I think we'll have to look to the future to see if any of them will emerge as an agent to be developed further.